Welcome back, students. Today we are discussing the last part of chapter 5. In today's topic, we will see how genes control the phenotype and also how a phenotype may be an outcome of different interaction rather than a mere one-to-one relationship. And these topics are Pleiotropy and Polygenic inheritance, showing some other pattern of inheritance and genetic expression. Up till now, we were discussing that a particular gene will express a particular trait. That means a pair of genes will show a particular phenotype or effect. But polygenic trait and pleiotropy shows some other pattern of inheritance. In polygenic traits, there are many genes contribute to a single effect, right? Whereas in pleiotropy, a single gene has multiple effects. That means in polygenic trait, many genes or multiple genes will show a particular phenotypic characteristic or a trait. Whereas in pleiotropy, one single gene will show many phenotypic characteristics or traits. So we will remember polygenic trait means many genes showing one effect, and pleiotropy means many effects that are shown by one gene. So we will see what is polygenic inheritance in detail. All the traits studied by Mendel have two alternative forms without any intermediate form in between. For example, if seeds we consider they were either round or wrinkled. If stem height, if we consider it was tall or dwarf, right? If purple flowers or white flowers. So there were two variants for a particular trait, right? And there were no intermediate variant in these traits. That means such traits were controlled by single pair of genes. But in polygenic inheritance showed many gradation, or we can say many intermediate variants for a particular characteristic. And that was because of many genes contributed for that trait. Example, skin color. So, polygenic inheritance is the inheritance of quantitative traits or the inheritance in which dominant polygenes have cumulative effect. That means it is the quantitative traits wherein many genes are contributing for a particular trait, or the inheritance in which dominant polygenes, that means many genes and that are dominant, having a cumulative or in total the effect is seen in an individual. So here the trait is controlled by additive effect of three or more pair of genes, and therefore it is called as polygenic trait. Such inheritance is called as quantitative inheritance because of multiple genes interaction. Let us assume that there are three genes A, B and C that controls the skin color in human. The dominant forms are capital A, capital B, capital C for dark skin, and recessive forms are small a, small b and small c for fair skin. The genotypes with all the dominant alleles capital A, capital B, capital C will be the darkest skin, and the recessive alleles small a, small bb and small cc will have lightest skin color. So as expected, there will be intermediate skin color too, as we see in F1 generation. So when cross is made between intermediate skin color, we see that there are numerous different shades of the skin color which will be seen. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. So, the presence of more dominant alleles of these polygenes will make the trait more prominent. The inheritance of human skin color was first studied by C.B. Davenport in 1913. The skin color is based on the quantity of melanin pigment and is believed to be controlled by at least 3 pairs of polygenes. So therefore we say it is the cumulative effect of multiple genes. The other example is human height and eye color. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment, giving rise to quantitative traits in the human. The next pattern of inheritance is pleiotropy. It is the ability of a gene to have multiple phenotypic effect or expression. That means here the single gene is responsible to show many phenotypic characteristics or traits. Such a gene is called as pleiotropic gene. Since here many traits are expressed by a single gene, such pattern of inheritance is also called as qualitative inheritance. One of the example is sickle cell anemia, that is the autosomal linked recessive disease caused due to mutation on a gene called beta globin. We have already studied in the topic human genetic disorder regarding sickle cell anemia, that is caused due to point mutation wherein an amino acid, glutamic acid, is replaced to valine at the 6th position of beta globin chain of hemoglobin. So here, due to mutation of one gene or mutation in a particular sequence, many multiple traits or phenotypic expressions are seen in the form of symptoms. We know that due to low oxygen tension, the normal RBCs become sickle shaped, and because of inflexibility, the sickle shaped cells or RBCs are not able to easily pass through narrow capillaries and thus have tendency to clot, and the degeneration leads to anemia. They get stuck in the capillaries and reduce circulation, leading to heart attack. Poor circulation and anemia deprive the tissues of oxygen, and therefore it leads to weakness, headache, muscle cramps, jaundice, liver failure, as well as blindness, and also affects the kidney. So such related symptoms or effects by a single gene, together we call it as syndrome. Yet another example is starch synthesis in pea seeds, which is controlled by a gene capital B. starch is synthesized effectively by capital B, that is homozygous. So therefore here large starch grains will be produced. Whereas in contrast, small b homozygous will have lesser efficiency in starch synthesis, producing smaller starch grains. But here capital B, small b, which is heterozygous, they show different phenotypes with respect to the synthesis of starch grains. So here B gene shows pleiotropy, that is, it is not only expressing for the starch synthesis but also the shape of the seeds as well as size of the seeds and starch grains. Here we see that capital B are the round seeds and bigger in size, whereas small b are the small seeds and they are wrinkled. Whereas here intermediate, that is heterozygous capital B, small b, they are of intermediate size, but since here capital B is there, therefore they are called as round. But otherwise B gene is showing the three different phenotypic expression, thus having more than one effect of a gene on the phenotype. So this was all about pleiotropic gene and polygenic inheritance. I hope you all must have understood all the topics that were discussed in this chapter, principles of inheritance and variation, and the important topics that were discussed for NEET preparation. We had also discussed about various problems that can be asked and also about the MCQs which may be asked for board as well as the NEET exam. Thank you very much.